Hey guys, this is Mrs. Hallman. This is part two of the Cancer and Stem Cell lecture for the Cell Division chapter. We last left off talking about Henrietta Lacks and how her stem cells have contributed, or I'm sorry, how her cancerous cells have contributed towards major scientific advances. If we proceed along, there are a couple other malfunctions within the human body that will lead to cancer, such as accelerator genes, brake genes, and lastly, we'll talk about P53, which is kind of a big and important one for you to know about. So let's start with accelerator genes. Accelerator genes are genes that would normally trigger our cells to grow and divide. So when a growth factor or some sort of signal or ligand lands on the right receptors, the signal transduction pathway will trigger the nucleus to enter into mitosis and to divide. When there's a mutation of those accelerator genes, we call it an oncogene, and that term is usually associated with the development of cancer. Because if we trigger the cells to grow and divide out of control without checks, that's going to develop cancer. And two really famous oncogenes are SRC and RAS. They're abbreviated for their gene loci. The second way we can develop cancer is if we have a brake gene that becomes mutated. So we have certain tumor suppressor genes that are meant to kind of keep the cell division cycle in check as far as speed goes. So if a cell is dividing out of control and it's not going through G1, S, and G2 with the proper amount of development and growth, these brake genes will basically pause the cell from dividing until the cell has passed the second checkpoint. If we have a mutation in this type of gene, of course, that will cause the cell to start to divide out of control in that faster speed without being checked with the brake genes. Third type of gene, P53 gene, is rather famous and it's extremely common. I think it's like half of the cancers out there, half of the diagnosed cancers now involve P53. So much so that it has been called the guardian gene. So normally functioning P53 is an amazing gene that's basically a proofreader or kind of like an inspector that after we go through the synthesis of DNA, it will scan the genome and make sure that there isn't any damage in the DNA. If there is, P53 has the ability to incite an editing mechanism where we'll actually go through and repair that genome. But if the damage is too great, the P53 gene can also trigger the cell to go into apoptosis or to basically eliminate the cell. The P53 can also arrest cell growth in the G1 stage. So if it doesn't pass that checkpoint, it will basically just pause that cell. So you can see plenty of applications for P53, a great gene to have around. The disadvantage is that when a mutation occurs in P53's gene on the chromosome itself, it renders P53 kind of useless. So it disables the cell death program and allows cancer to start to spread rapidly. So this image shows exactly that. It shows the normal P53 gene up here, that when that is released in the cell and the cell goes through division where there is mutant DNA, the P53 gene has the options of either fixing it or prompting the cell to go through apoptosis. When P53 is non-functional or mutated, and those same DNA mutations arise, then if there is a malfunctioning P53 gene, then that will lead to uncontrolled cell division in cancer. Same idea in this image. It basically says that the P53, when damage occurs to that particular gene, the cell cycle is going to continue out of check and the cells can become cancerous. Normally functioning P53 can either arrest the cell cycle, repair the DNA, or decide to restart that cell cycle. If none of those things work, then it can go through apoptosis. So it helps protect against those cancerous cell developments. One of the really revolutionary ideas and finds of this century was the discovery of telomerase. So we're very comfortable with this idea of chromosomes looking kind of like an X. And you know that one of those chromatids, one half of that X formation, is one long strand of DNA kind of all wound around. And we talked about how that DNA organizes itself. We're going to learn later in our DNA chapter that when we replicate DNA, we actually lose a little tiny, tiny portion of the end of that strand of DNA from the very, very end of that chromosome, which which actually means that every single time that your cells divide, your chromosomes get a little bit shorter. We're talking really, really short amount, but we think that that might make somewhat of a difference. So you can kind of think of it kind of like if you have a pair of shoes, there's a little plastic edge around the very end of your shoelaces that prevent the shoelaces from fraying over time. You can kind of think about the telomere as being that. Typically, we don't have any coding genes in that region. It's an area that we're still to this day discovering what is actually stored in that genetic information. But telomerase is a gene that can be turned on that will actually rebuild telomeres. So what that means is that even after the cell divides and we lose that little area of the DNA, that little stretch of DNA, the telomerase will actually rebuild it. So it's as if that cell hasn't divided. And cancer cells can actually turn on that gene, which helps describe why those cells can divide out of control so many more times than our normal cells. Another mechanism that's really common when we see tumor developments is this process called angiogenesis. And if you break that word down, biologists are lazy when we name things. We name things after exactly what they say. So genesis means to create or to begin, and angio is a term that's often used to refer to blood vessels or veins. So tumors, just like other normal cells, need nutrients. They need to get rid of wastes. They need to maintain homeostasis, and they need to go through the exchange of nutrients, just like normal developing cells. And the quickest way to do that, of course, is to attach those cells or to bring them closer to a bloodstream, because we know the blood is the circulatory highway of our cells that will deliver nutrients and get rid of wastes. So tumors have the ability to maniacally recruit blood vessels to actually vascularize their tumor so that those tumor cells can be healthier, get more nutrients, and grow even larger. One example of this, and I believe there's several other different versions of tumor growth factors, is VEGF, which is vascular endothelial growth factor. And if you look here, there's a pre-existing tumor that is not yet vascularized, so it doesn't have a blood vessel near it, and it will secrete this growth factor that will trigger the endothelial cells in this area to actually expand and migrate closer to the tumor, and eventually it will create a microvascular channel that will supply that tumor with more blood and more nutrients. There's a very, very strong tie between heredity and cancer, which makes sense because cancer has a lot to do with gene mutations, and obviously heredity is how you get those genes. And colon cancer is kind of a tragic but really good example of how cancer can be inherited. So colon cancer is a type of cancer that can actually accumulate mutations. So you can see up here on this side of the screen, we have a normal colon cell that progresses the whole way down to a metastasized cancerous colon cell. And over several generations of people and also of that cell going through divisions, it can accumulate multiple mutations. So it can be demethylated, it can have a mutation in the RAS gene, which we talked about earlier. It's a growth factor. We can have the loss of the P53 gene that would finally allow that cell to become cancerous. So colon cancer is one of those cancers that can actually develop those mutations over time before it becomes an actual dangerous cancer. That, of course, can be passed on to multiple generations, unfortunately. Another cause of cancer, which was probably a little bit more well understood in the past 50 years once we've really delved into virology, is cancer that's caused by viruses. And one particularly relevant virus that does cause cancer is the human papillomavirus, which is known as HPV. And there are a bunch of different strands of HPV, but it has been known to cause cervical cancer. So there are certain versions of HPV, so HPV-6 and HPV-11 are shown here, will cause the normal development of that virus. But there are a couple cancer-causing types, such as HPV-16 and 18, that will lead to an increased risk of cervical and vaginal cancer.